In this video, we're going to provide a brief overview of cellular respiration. We can get into the nitty-gritty details later, but for now, we want to give you the big picture. We've already discussed the ATP cycle and the fact that cellular respiration is an exergonic process that provides energy that is used to regenerate adenosine triphosphate from adenosine diphosphate and an inorganic phosphate group. We'll mostly be discussing aerobic respiration, which is a type of cellular respiration that requires oxygen. Let's also recall our simplified formula for cellular respiration, which is glucose plus oxygen, reactive form carbon dioxide, and water. Now, there's a lot of details kind of simplified here, but for now, this is our overall reaction. Recall that this is an exergonic process because we're breaking down, or in other words, catabolizing glucose. Energy is released by the process of cellular respiration, and we couple the exergonic process of breaking down glucose or other organic molecules to the endergonic process of regenerating ATP. Aerobic respiration can be divided into four main steps, which are glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. There's a lot of detail on this slide, but don't worry, we're going to go through this step by step and break down the inputs and outputs of each of the four phases of cellular respiration. We've already discussed the last phase, which is oxidative phosphorylation, including how transferring electrons from molecule to molecule in the electron transport chain provides the energy to maintain the hydrogen ion gradient that is essential for chemiosmosis, which is the process of hydrogen ions diffusing down their gradient through ATP synthase, which catalyzes the reaction that produces ATP. Hopefully by now that sounds familiar. Now we're going to start diving into the first three phases of cellular respiration: glycolysis, pyruvate oxidation, and the citric acid cycle. In these phases, high-energy electrons are harvested from glucose or other food molecules and shuttled by electron carriers NADH and FADH2 to the protein complexes of the electron transport chain. So that's really the big picture and the main point of this whole process, to provide the energy that powers the electron transport chain so that chemiosmosis can continue to take place. All right, so our electron carriers, NAD and FAD, these are molecules that can easily accept electrons or release electrons. Let's look at the major electron carrier. Again, nicotinamide adenine dinucleotide, or NAD. This molecule can exist in an oxidized form, in which we call it NAD+, or a reduced form, in which we call it NADH. Which form has more electrons? Hopefully you answered NADH, the reduced form. NAD+ can gain electrons, meaning it is being reduced to become NADH. This happens when NAD+ accepts electrons from the organic molecules being broken down in the process of cellular respiration. And NADH can be oxidized. In other words, it can lose electrons. This happens when NADH transfers the electrons that have been harvested from organic molecules to complex 1 of the electron transport chain. Recall, this is a redox reaction. NADH is being oxidized to NAD+, and complex 1 is being reduced as it accepts those two electrons. Similarly, our other electron carrier of aerobic cellular respiration, flavin adenine dinucleotide, or FAD, can exist either in an oxidized form, shown on the left, or a reduced form, shown on the right, which has 2 more electrons than the oxidized form. And it has a similar function as NAD. It harvests high-energy electrons and delivers them to the electron transport chain, in this case to complex 2. These electrons that are delivered by NADH and FADH2 are passed through the electron transport chain to oxygen, which is the final electron acceptor. Remember that oxygen can pick up 2 electrons, also 2 hydrogen ions, and combine to form water, which is one of the major outputs of cellular respiration. Some of the energy released by electron transfer in the electron transport chain is used to pump hydrogen ions against their gradient from the mitochondrial matrix to the intermembrane space. This creates our nice hydrogen ion gradient, and these hydrogen ions can then diffuse down their gradient through ATP synthase, which releases the energy necessary to build ATP from ADP, an inorganic phosphate. Okay, so where do these processes take place in the cell? Let's zoom in and take a closer look. The first step, glycolysis, takes place in the cytoplasm of the cell. All cells perform glycolysis, and it results in the net production of 2 ATP molecules. It also results in the 6-carbon glucose being broken into 2 3-carbon pyruvate molecules. Pyruvate is then transported into the mitochondrial matrix. Here is the structure of the mitochondria. Mitochondria have 2 membranes, an outer membrane and an inner membrane. Remember how mitochondria are evolutionarily derived from bacteria? Well, the inner membrane is the original bacterial cell membrane, and the outer membrane is a remnant of the vesicle that engulfed the ancestor of mitochondria. The space between the 2 membranes is, very logically, called the intermembrane space, and the space inside the inner membrane is called the mitochondrial matrix. Many copies of the electron transport chain are embedded in that inner membrane. All right, so pyruvate oxidation takes place in the mitochondrial matrix, and the process is pretty much what the name suggests. The 3-carbon pyruvate molecules are oxidized. In other words, pyruvate loses electrons which are accepted by NAD+. That reduces NAD+ to NADH, and it can now carry those electrons to the electron transport chain. In the process, each pyruvate is converted to a 2-carbon acetyl group, which can then be used in the citric acid cycle. Where does the extra carbon go? Well, it leaves as carbon dioxide and is able to diffuse out of the cell. The citric acid cycle completes the process of glucose catabolism. By the end of it, all of the original carbons from glucose have become part of carbon dioxide molecules. And all of the extractable energy from glucose, which is in the form of electrons, has been accepted by NAD+ and FAD, reducing them to NADH and FADH2. These electrons, once again, can be delivered to the complexes of the electron transport chain, which maintains the hydrogen ion gradient that allows the bulk of ATP synthesis in the cell to take place.